I am Syed Wajiha Tahsin, Department of Biochemistry, SRR and CVR Government Degree College, Vijayawada. Today we are going to discuss about qualitative analysis of carbohydrates. Students, in the first semester you have learned the theory part of carbohydrates. You have learned about the structure, properties and biological importance of carbohydrates. Today we are going to discuss how to analyze these carbohydrates, how to find out which carbohydrate is present in the given solution, and also to find whether the given solution contains carbohydrate or not. So, for this first of all we will go and I will introduce you about the carbohydrates. So, how they are classified, what is the biological importance, then we will go into one by one the analytical tests. So, we will start. So, what is the aim and objective of today's lecture? An unknown sugar carbohydrate, unknown sugar or carbohydrate by carrying out a series of chemical reactions and also to determine few properties of carbohydrates. Coming to the general introduction of carbohydrates. So, what are the carbohydrates? Carbohydrates are nothing but they are polyhydroxy aldoketo sugars and their derivatives. As you know that carbohydrates are present in all living organisms. They exist where they are playing very important structural and as well as metabolic role in all living organisms. In all carbohydrates, you know that sugar is the most important carbohydrate which is playing the role of source as a source of energy. It is a major source of energy in many living organisms. Then you will this glucose is a very important compound and many metabolic products are also formed from glucose, and all the carbohydrates in animals are also synthesized from glucose. So, coming to how to classify these carbohydrates. Carbohydrates are classified as monosaccharide, disaccharides or oligosaccharides and polysaccharides, basing on whether on hydrolysis they give monomer or dimers or polymers. So, monosaccharides are the one which on hydrolysis give only a single carbohydrate or single monomer. That is the simplest compounds. So, coming to disaccharides, they on hydrolysis give 2 monosaccharides. Oligosaccharides are the one which on hydrolysis give 2 to 10 monosaccharides, and polysaccharides are the one which on hydrolysis give many monosaccharides. Coming to what is the biological importance of monosaccharides. So, monosaccharides are playing very important biological roles such as if you take trioses. So, examples of trioses are glyceraldehyde, dihydroxyacetone, and coming to tetroses you have erythrose, erythrulose, and coming to pentoses you have ribose, arabinose, xylose, ribulose, and coming to hexoses you have glucose, galactose, mannose and fructose. Like that you have many different varieties of monosaccharides. So, this classification is based on number of carbon atoms. If you have 3 carbon atoms, then they are called as trioses. If you have 4 carbon atoms, then they are called as tetroses. If the number of carbon atom is 5, then it is called pentoses. If you have 6 carbon atoms, then they are called as hexoses. Then disaccharides, if you take disaccharides which on hydrolysis give 2 monosaccharides, among them the isomaltose. So, isomaltose is the one which on enzymatic hydrolysis of amylopectin, which is the component of starch, it gives on hydrolysis isomaltose, and maltose is produced from hydrolysis of amylose. Lactose is the one which is present in milk. It is present up to 5% in different types of 3 to 5% in different types of milks. Then coming to sucrose, which is the major component of cane and many fruits, vegetables, etc. Then polysaccharides. These polysaccharides are usually classified as homopolysaccharides and heteropolysaccharides. Homopolysaccharides are the one which on hydrolysis will give similar type of monosaccharides. Heteropolysaccharides are the one which on hydrolysis give two or many varieties of monosaccharides. So, polysaccharides can be also classified as structural polysaccharides and storage polysaccharides depending on their biological role. So, here we have some of the examples. Homopolysaccharides, if glucose is the main unit of homopolysaccharide, then they are called as glucosone. If fructose is the main unit, then they are called as fructosone. If galactose is the main unit, then they are called as galactosone. So, here you have glucosones examples: starch, glycogen, dextrin, cellulose, and fructosone you have inulin, which is present in the root of dahlia. Next, galactosone, which is example is agar. Then coming to heteropolysaccharides, they are again classified as non-sulfated, sulfated and neutral polysaccharides. Non-sulfated heteropolysaccharides example hyaluronic acid, chondroitin, and sulfated heteropolysaccharides you have keratin sulfate, chondroitin sulfate, dermaton sulfate, heparin, and neutral polysaccharides are blood group substances. Then we will start how to analyze these carbohydrates. So, students, first introduce yourself.
I am K. Vijay Krishna, student of Biochemistry in SR and Xavier Government Degree College, Vijayawada.
I am Tulasi Surya Sindhu M. I am student of Biochemistry in SRR and Xavier Government Degree College, Vijayawada.
I am M. Ratnakumari. I am student of Biochemistry, SR and Xavier Government Degree College in Vijayawada.
I am K. Bhavani, student of Biochemistry, SRR and Xavier Government College in Vijayawada.
I am Arshi. I am a student of Biochemistry and SR and Xavier Government Degree College, Vijayawada.
I am student of G. Shantakumari, student of Biochemistry in SR and Xavier Government Degree College, Vijayawada.
Students, now we are going to prepare sugar solutions. 1% of sugar solutions, how you can prepare? By dissolving 0.25 grams of sugar in 25 ml of water, we can prepare 1% of sugar solutions. So, we have prepared monosaccharide, disaccharide and polysaccharide sugar solutions. So, you have glucose solution here, and you have fructose solution here, and you have ribose solution, pentose solution here, and you have sucrose, maltose, lactose, disaccharide solutions, and starch, polysaccharide solution. So, one by one we will go into how to analyze. So, these sugars, first of all we will start with identifying whether the given solution is carbohydrate or not. So, for that the test we used is Molisch test. In Molisch test, what we are going to do, we will take the sugar solution by any one of the carbohydrate solution, and also we are taking control. So, for each test we have to take control and test solution, so that you can be very clear whether the reaction has happened or not. So, in the control solution we are just adding the water, and in the test solution we are adding the carbohydrate solution, right. So, first we will do Molisch test. So, Molisch test is used to detect whether the given solution is carbohydrate or not. To perform this test we need Molisch reagent and H2SO4. So, what is Molisch reagent? Molisch reagent consists of ethanolic solution of alpha naphthol, and when you add this Molisch reagent 2 to 3 drops to the given solution, and you have to add H2SO4 by keeping the test tube in slant position. Slowly, drop by drop, you have to add H2SO4. Then what happens? What is the reaction really went on? So, when you add H2SO4, H2SO4 causes the dehydration of sugar and it gives furfurols and furfurol derivatives. Furfurols or furfurol derivatives are formed. These furfurol derivatives is condenses with this alpha naphthol. You have in Molisch reagent, you have alpha naphthol, condenses with alpha naphthol to give violet or brown color ring which exists between the two liquids, at the junction of two liquids. So, first we will perform this test. You carefully observe and one of you has to perform the same test. So, first of all we are taking water as the control. So, we are taking 2 ml of water in the test tube, right. So, this is used as a control. So, better if we do the test in small test tube, so that you can observe the formation of ring at the junction. Okay. So, we will perform the test in small test tube. So, your ring will be very clear. Cam wipe. Okay. So, first of all in the control we are adding 2 ml of water. Then this is the control, one of you hold, and this is the test. So, any one of the carbohydrate you can take as a test. Okay. So, any one of the carbohydrate. So, now we are taking the glucose. So, you have to pipette out. The glucose solution. Then you have to add 2 to 3 drops of Molisch reagent. Okay, this is the Molisch reagent. So, we are adding 2 to 3 drops of Molisch reagent to the test solution. This is test. In the same way, you just hold this one. Now, I am adding the Molisch reagent to the control. Okay. Then, what is the next step? We have to add H2SO4. So, this is a concentrated sulfuric acid. So, we are adding the concentrated sulfuric acid to the control by keeping it in a slant position, right. So, like that we have added, right. So, then see, this is the test solution. So, we are adding the concentrated H2SO4 by keeping the test solution in slant position. So that you can observe a clear ring at the junction of two liquids. Okay. Now, students, see, can you observe any ring here, brown color ring? Yeah, it is there. See, observe. So, slowly it is forming, slowly it is forming. So, what you are observing? Brown color ring between the junction of two fluids, right. So, here if you see in the here you can't see any such type of ring. So, see clearly, okay. Can you discriminate?
Yes.
Yeah. This is the control and this is the test. In test you can observe the brown color ring, okay. It indicates the given solution is a carbohydrate, right. So, then what is the principle behind this? Once again you revise what happens when you added H2SO4. H2SO4 causes the dehydration of the sugar solution, sugar, to form furfurol and furfurol derivatives. These furfurol derivatives reacts with alpha naphthol, condenses with alpha naphthol to give violet color ring. So, this is a confirmation test for carbohydrates. Then in the next step, so already you have introduced that carbohydrates are classified as mono, di, polysaccharides, and carbohydrates are also classified as reducing and non-reducing. So, do you aware of what are reducing and non-reducing? reducing sugars are the one which are having free anomeric carbon. If the functional group is free and ready to react ready to participate in different types of reactions, then it is called as reducing sugar. If the functional group is not free and whether if it is involved in linkage, then it is called as non-reducing sugar. Okay. So, like that you can discriminate reducing and non-reducing. So, in theory what you have read, so what are reducing sugars? Reducing examples for reducing sugars: glucose, fructose, maltose, okay, lactose, all these are reducing sugar. Coming to non-reducing sugar, sucrose is the non-reducing sugar. Why sucrose is non-reducing? Because in sucrose, glucose, you have glucose and fructose. Sucrose is a disaccharide made up of glucose and fructose, in which the first anomeric carbon of glucose is first carbon and anomeric carbon of fructose is the second carbon. Both are involved in linkage, glycosidic linkage. That is why in sucrose you cannot find any free anomeric carbon, right. So, now we are going to perform this test by taking 3 test tubes. One is non-reducing sugar, second one is reducing sugar, and the third one is control. Okay. So, we will perform the test which is called as Benedict's test. Now, you please observe. Okay, Vijay, we have already performed the Molisch test. Yes, now you do.
Okay, ma'am.
So, take this control and this is the test.
Okay,
so, what you have to add in control?
Water, ma'am.
Yeah, take water,
Yes.
take 2 ml of water, add, add 2 ml of water, good. Next, take this one, this is the test. So, now what you have to do?
Carbohydrate solution, ma'am, that is
do not use the thumb finger, always use the third finger, right. For pipetting you should use this finger, not the thumb finger, right, so that you can easily move, right.
yes, ma'am.
Then this is your control.
Yes, ma'am.
So, what you are going to add?
Molisch reagent, ma'am.
Yes, take carefully. How many drops you have to add?
2 to 3 drops, ma'am.
Yes. Pipette it. Yeah. Then take this is test. To test also you have to add
2 to 3 drops, ma'am.
Right. Then what you have to do?
You have to add concentrate H2SO4, ma'am.
Yes. Take the control.
In slanting position, ma'am.
You have to keep it slanting position. Yeah, no problem, suck it down, right. Keep it in more slanting position. Yes, add. Okay, stop, right.
No ring is observed.
Do you observe any ring here?
No, ma'am.
But you can observe the 2 fluids, okay.
Yes,
So,
ma'am.
there is a junction, here junction you can't see any brown ring. So, this is the test.
In slanting position, ma'am.
Add slowly, drop by drop.
Yes, ma'am, we got, ma'am.
Right. So, can you observe any ring here? Brown color ring. So, it
Yes,
takes some
ma'am.
time to form. So, it is forming slowly, right. So, brown color ring you can observe. Yes.
ma'am.
Okay.
We got, ma'am.
So, from this we can discriminate that this is the given solution is what? Carbohydrate, right. Then we are going to perform the next test. Okay. The second test we are going to do is to discriminate whether the given sugar solution is reducing or non-reducing. Students, as you learn in theory that the carbohydrates are also classified as reducing and non-reducing. So, what are reducing sugars? Reducing sugars are the one which are having free anomeric carbon, that is the functional group is free, so that it is ready to interact with other chemical reactions. So, if in reducing sugars you have the free anomeric carbon or free functional group. In non-reducing sugars the functional group is involved in linkages. So, it is not free to react with in other reactions. Okay. So, now we are going to discriminate whether given solution is reducing or non-reducing. So, as you know that the most reducing sugars examples are nothing but glucose, fructose, coming to disaccharides, lactose, maltose. In all these you have one free anomeric carbon. Coming to sucrose, if you take, which is considered to be as non-reducing sugar. Why it is non-reducing? Because in sucrose you have glucose and fructose. In glucose the first carbon, that is aldehyde group, is, and in fructose the second carbon, that is keto carbon. Both are involved in linkage. Okay. That is why there is no free anomeric carbon. So, sucrose does not give positive test. Now, whatever test we are going to perform. So, for determining whether the given solution is reducing or non-reducing, we are going to perform the Benedict's test. Okay. So, this is the Benedict's reagent. This is the Benedict's reagent, and this reagent consists of copper citrate. You have citrate in this, sodium citrate is there, and you have sodium carbonate, and you have copper sulphate. In copper sulphate the copper ion is existing in 2+ state, that is cupric state. So, what happens when you perform this test? So, if you perform this test with reducing sugars, as the reducing sugars are having free anomeric carbon which is involved in the reaction, which is aldehyde or keto group, and this group is oxidized to carboxylic group. This group is oxidized to carboxylic group, and copper that is present in Cu2+ form, which is converted into Cu+ form, that is cupric form is converted into cuprous form. So, what is the reason for this means? So, here what is the principle behind this? So, in alkaline conditions usually the monosaccharides or disaccharides, that means reducing sugars, they form enodiols. By tautomerization they form enodiols. These enodiols are highly reducing agents. Okay. So, that is why they will reduce the copper which is existing in 2+ form into Cu+ form. So, copper get reduced from Cu2+ to Cu+ state, and at the same time the carbohydrate gets oxidized from aldehyde keto group to carboxylic group. Okay. So, glucose will give gluconic acid, So, this is the Benedict's test principle. Now, I will perform how to do this test. Okay. So, you have to take 5 ml of Benedict's reagent. Okay. To this 8 drops of the sugar solution should be added. Okay. When you add the Benedict's reagent and sugar solution, then you have to heat. Okay. Here we are using the spirit lamp. So, spirit lamp, in the flame of spirit lamp you are going to heat. So, depending on the amount of carbohydrate present, depending on the amount of reducing sugar present, you can observe 2-3 different range of colors. If the carbohydrate is present in less amount, then it gives green color. If it is present in moderate amount, it gives a orange color. If it is present in high concentration, then red color is observed. So, why this red color? Because I already told you that copper is converted into Cu+ form. So, Cu+ form, that is copper reacts with hydroxide ions to form copper hydroxide. This copper hydroxide on heating, it gives the red color. Okay. So, that is the principle behind this test. Clear? It is clear for you. So, what happens? Enodiols are formed. Enodiols are highly reducing agents. These enodiols they will convert the Cu2 plus into Cu plus form. Cu plus form interacts with OH minus ions to form copper hydroxide, which on heating gives red color. Okay, that is the principle. So, let it ma. So, this test we will perform by using 3 different test tubes. One is taken as control, another is for reducing sugar and another is for non-reducing sugar. In control, we are just taking the water. To this we have to add 5 ml of Benedict's reagent. Say control, you hold ma. And this is the test. Here we have to take the glucose solution. Eight drops means a small amount of glucose solution or reducing sugar, then you are adding the Benedict. Okay. So, you hold, then we are taking the sucrose. Sucrose is the non-reducing sugar. So, it gives negative test. So, you are adding 8 drops or small amount of sucrose. To this we are adding the Benedict. Okay. So, this is non-reducing sugar, right. First I will perform with non-reducing, then we will go for the control. On heating, you observe. Heat for half minute or 30 seconds. So, you just count 1, 2, 3 like that. If you count for 30 seconds, so can you observe any change in color? No. So, then you take the control. So, this is the control. Okay. Can you observe any color change? No. Now, we are performing with our test solution. Test is, we already know that this is a reducing sugar, okay, because we have taken the glucose. Can you observe the color change? Yes. So, it is observed to be green. Okay. So, because of small amount you observe the green color, and later on if you heat it, it is converted into red. Okay. So, can you observe the red color?
Yes.
As we have taken. Okay. So, red color, right. So, this is the, so can you compare these two? Okay.
Yes,
Yeah. So, this is the control and this is for sucrose, that is non-reducing, okay, and this is the reducing. So, this is the Benedict's test. Okay. You please come, Santakumari, perform the same test. Take 3 test tubes. So, give to take the water as control. Okay. Give the control to him, hold it, right. Next, take the sample. First take the sucrose, which is non-reducing, right. Yeah. Okay. Right. And this is the glucose. Okay. You keep all this for washing. Oh, be careful. Okay. Then add the Benedict reagent, 5 ml to each. Yeah, okay, enough. Add carefully. This is the test solution. Now we add it to control. This is sucrose, non-reducing. Second, no problem. Second. Okay. Right. Right. Then control. No problem if the test tubes are exchanged, also no problem, because while you heat you will identify, right.
Sure.
Okay. Keep it back, right. Then one by one you start heating. ఇక్కడ పెట్టమ్మా, రెడ్ మీద కాకుండా కింద పెట్టు. Right, half minute is enough. Okay. Then, so it is non-reducing. Hey, you hold this. Good. So color is changed, right? Okay. Okay. So green to red. Okay. You can observe. So this is the test solution, which is a reducing sugar. Now that is the control. No color change, right. So within half minute you can't observe. So from this you can clearly identify which is a reducing sugar. This test is usually carried out in clinics for urine analysis also, whether the patient has suffering with diabetes or not, okay, because diabetic patients in urine consists of glucose, right. Next we are going to perform the next test, that is Barfoed's test. So this test is to discriminate whether the given solution is disaccharide or monosaccharide. So, now up to this what you have performed, you find out whether the given solution is carbohydrate or not. In the second step, you found out whether the given one is reducing or non-reducing. Then now you are going to commence the test to distinguish whether the given solution is a monosaccharide or disaccharide. Okay. So, monosaccharides are the one which has only one monomeric unit, disaccharides which on hydrolysis give 2 monomeric units, 2 monosaccharides, right. So, how this test is performed and what is the principle behind the test? This is the Barfoed's reagent. This is the Barfoed's reagent. So, Barfoed reagent. So, here in this test also you have the principle little bit similar to the Benedict's test. What is the principle? How? Why? What is the reaction goes on? Why the color change observed? So, here in Barfoed's reagent you have glacial acetic acid and copper acetate. This copper acetate is converted into cuprous oxide, copper oxide. So, in copper oxide the copper is existing in Cu plus form. In copper acetate you have the copper existing in Cu2 plus form. Okay. So, to Cu2 plus form to Cu plus form, that is nothing but reduction of copper it takes place. Okay. So, this test usually what you observe in this test, the copper acetate which is existing in blue color, it is converted into copper oxide which is existing as a red color precipitate, So, this when we perform this test, if we heat with the sample solution for 1 minute, if the test comes positive, that means you can observe the formation of precipitate, red color precipitate within 1 minute, it indicates it is a monosaccharide. If you observe the formation of precipitate after prolonged heating, it indicates it is a disaccharide. Right. So, we will start. So, 2 ml of Barfoed's reagent and 2 ml of sugar solution has to be taken in the test tube. Okay. So, after taking, mix well and then heat it. So, if you can observe the formation of precipitate within 1 minute, then it is indicated as monosaccharide. On prolonged heating, if you can observe the formation of red precipitate, then it is considered to be as disaccharide. So, for example, you have here monosaccharides and disaccharides. So, we are taking one of the monosaccharide. So, you can take glucose, either glucose or fructose. Okay. So, you have ribose also which is a monosaccharide. So, we are taking this glucose. Here also I will perform this for control, test and for disaccharide. Okay. So, we are taking as this into control for monosaccharide and for disaccharide. So, first control, we are going to add okay. So, this is control, we have added 2 ml of water, control, one of you hold, and then we are going to add monosaccharide. Okay. So, I think that is washed, give one of the washed. So, this is 1 ml pipette, that is why I am adding 2 times. Okay. So, this is the test. Maharshi, you hold the test. Then keep it for washing. Then give this one. Then we are going to add the disaccharide. Okay. So, we are taking maltose here. So, 2 times you have to add as it is a 1 ml pipette. Okay. Right. Then we have to add the Benedict Barfoed's reagent, sorry, Barfoed's reagent. So, how much it is? We have to add
2 ml.
Usually you have just mix the once the reagent bottle, okay, so that when anything gets precipitated. So, 2 ml of Barfoed's reagent to maltose, right. This is the disaccharide. Next this is the monosaccharide. Now I am adding for monosaccharide. Hold it. Now I am adding to control. Then you have to heat this. This is control, okay. Can you observe any change in control? Just guess.
No, ma'am.
No. Because there is no carbohydrate, that's why
There will be no reaction.
no reaction. So, boiling started. Can, if you listen to that sound, it indicates boiling started, okay. Okay. Then this is the control. This is disaccharide. So, can you observe any color change within 1 minute?
No, ma'am.
Okay. So, then you hold it and do it the same thing with your test, that is monosaccharide. Okay. You just heat it, ma. Yes,
Color is
yes.
changing, no?
Yeah, color is changing.
Slowly.
You can observe the formation of precipitate
Within 1 minute, ma'am.
within 1 minute, right. In a short time you can observe the, if you cool it you can clearly see there is a precipitate at the bottom, okay. Can you observe? So, this is only because of the flame, okay. If you wipe, if you have any cotton, so take the cotton. So, we have to perform all these tests by using water bath, okay. Then it will be very nice. Okay, can you see, can you observe any precipitate here?
No,
At the
ma'am.
bottom? No, right. Okay. So, if you take this one and this one, okay, it is due to flame only. So, you can observe the formation of red color precipitate here, okay. Within 1 minute, it indicates that it is a monosaccharide. It indicates it is a monosaccharide, right. Then we will go for the next test. So, you have already performed, one of you has heated, right. So, then we will go to the next. Okay. Till then you have performed the tests which distinguished the monosaccharide and disaccharide. Now you are going to perform another test which will discriminate whether the given solution is aldo or keto sugar. Aldo or keto sugar. You know that sugars have functional groups. Carbohydrates are nothing but polyhydroxy, aldo or keto sugars. To discriminate the given solution as aldo sugar or keto sugars, we have to perform this Seliwanoff's test. Seliwanoff's test. So, Seliwanoff's test, in this you have Seliwanoff's reagent which is consisting of HCl and resorcinol. HCl and resorcinol. So, when you add Seliwanoff's reagent to the given solution, what happens? In it you have HCl. HCl will causes the dehydration of the sugar, and it gives rise to the formation of furfurols. These furfurols condenses with resorcinol to give cherry red color product. Okay. So, you can observe cherry red color complex is formed inside the test tube. Okay. So, one of you come and commence the test. So, take the test tube, add 5 ml of Seliwanoff's reagent. Better to take 5 ml pipette, ma?
Yes, ma'am.
So, it is there in Benedict's reagent. So, wash it. Okay. Okay. Right. Take 5 ml of Seliwanoff's reagent. Right. Yeah. So, this is a control, and take another test tube to which also you add 5 ml of Seliwanoff's reagent. Then for control what you have to add? You have to add water. Okay. 1 ml of water, just add 1 ml of water to the control. Okay. Then what is the keto sugar? Fructose. Fructose is the keto sugar. Now add fructose. Yeah. This is your test. I am holding the control and
sugar solution.
Sugar solution.
Which is a ketose.
Ketose, right. Then,
now
so,
I have to heat it.
Yeah. Okay. Now you just heat. Okay.
First
heat. Yeah.
I am heating the control to check whether any chemical reaction is happening without any sugar solution. Okay, just heat for 1 minute. So, heating
Can
is
you
over,
observe
but
any
no
change?
chemical reaction is
okay, no reaction. Then do the same with the test.
slowly color is getting altered.
Yeah. Okay. Hit the same this one also, so that you can observe what is the color change. For prolonged heating. Yes. 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 Okay. Right. So can you observe the color change, ma?
Yes,
so what observation you have done here? So this is the cherry red color, and this one does not give any color change. That means it is control and it is the test which is having the keto sugar. Right. So, once again explain the principle. Why this is giving the red color? Because Seliwanoff's reagent contains HCl and resorcinol. HCl, when you add the HCl to the test solution, what happens? The sugar is converted into furfural. Okay, furfural or furfural derivatives by dehydration of the sugar. These furfural derivatives condenses with resorcinol to form the cherry red color. Right. So, we have completed the aldo or keto sugar. Now we are going to perform another test to detect whether the given solution is a monosaccharide or polysaccharide. Monosaccharide or polysaccharide. So, you know that polysaccharides, what are the examples for polysaccharides? Starch, glycogen, right. So, starch, glycogen, inulin, and structural polysaccharides are cellulose. Right. So, for this we are taking the starch solution. Now, to that iodine is added. So, as you know that iodine does not react with starch, but you can observe a color change. So, what is the reason? Because iodine gets absorbed in the helical structure of starch. It forms coordinate complex with the helical structure of the starch and it goes and impregnated in the structure of starch. So, it gets adsorbed to the starch. Thereby, you can observe the refractive index of the solution changes. That is why you can observe a clear color change. Okay. So, you can observe a blue color. So, do it with the starch and control. One of you come forward. Okay. So, take the 2 test tubes. One is control, pour some water, 2 ml of water. Be careful. Right. So, this is the control and flame avutadamma. Be careful. This is the starch solution, right. So, I will mix it. So, keep it for washing. Then so, you directly take some amount of starch, right. Okay. Right. So, then add iodine, right. So, just a few drops of iodine is enough. So, to the control also I am adding the iodine. Hitting me. Right. So, see, can you discriminate the color change? So, what is the color here? Purple. Purple, blue color. Okay. So, this is a no color change, only iodine color is observed. Okay. So, this is a clearly indicates it is a starch solution. Okay. So, what is the principle behind this? Once again. So, the starch is nothing when you add iodine to the starch, it will goes and impregnate in the helical structure of starch. Thereby, the refractive index of the solution changes and the solution is observed to be in violet color. Okay. So, now you can distinguish between mono and polysaccharides, right. Mono, di and poly. Only polysaccharides will give the positive test with iodine. If you add glycogen, it gives the red color, starch gives the blue color. Okay. On hydrolysis of starch, you can observe 3 different components, but that is amylodextrins, erythrodextrins, acrodextrins. So, amylodextrins will give violet color, erythrodextrins will give red color, acrodextrins will give no color. Okay. So, now you are clear, right. Then we have to perform another test to discriminate whether the given solution is pentose or hexose. Okay. What is pentose? Which is having 5 carbon atoms. What is hexose? Hexose is having 6 carbon atoms. So, this test is performed by using ribose. Okay. So, we are taking the ribose and we are also taking the control. Right. So, one of you come forward. So, take the water, yeah, 2 ml of water, right. So, in this you take the ribose, directly you can pour small amount of ribose, right. Okay. So, this you have to add Bial's reagent. Okay. Now, stay for some time, I will explain how this reaction is goes on. So, when pentose is in Bial's reagent, what you have? You have same HCl plus arsenol. Here you have arsenal. In Seliwanoff's test, what you have? You have resorcinol. Here you have arsenol. So, when you add HCl and arsenol, HCl causes the dehydration of the sugar to form furfurols. These furfurols condenses with arsenol to give the colored complex. Okay. So, here you can observe a green color with pentoses. Pentoses will form the furfurols. Furfurols when reacts with arsenol, they will give the green color. Okay. So, now add Bial's reagent. This is the control, you have added the Bial's reagent, and this is the test. You have added the Bial's reagent, then just heat both and observe the change. So, now you are heating the control. Once you mix it, so that so mixing should be carried out like this. Okay, so that reagent and the sugar mixes properly. Boil for 1 minute. Okay. Now you heat this one, this is the test. So, there is a color change. So, why the color change occurs? So, it is happening. Why the color change is observed? Because what are the reagents in Bial's test? HCl and arsenal. Okay. HCl will dehydrated the sugar to form the furfurols. These furfurols condenses with the arsenol to give the green color. Yeah. It's changing into green color. Right. Okay. So, you can observe through the walls. Okay. You can observe the clear green color. Okay. Can you observe the bubbles? You can observe the green color, right? Okay. Yeah. So, can you discriminate the both? So, here there is no, yeah, this is the green color. It indicates the presence of pentoses, pentose sugars, right? Till now, what you have done, so you have taken the sample solution. So, first of all, you have to find out whether it is a carbohydrate or not. For that, you have done the Molisch test. So, if brown color ring is observed, you can you can conclude that it is a carbohydrate. Then you have performed Benedict's test, which discriminates the reducing and non-reducing sugars. Then you have performed the Barfoed's test, which is used to distinguish the mono and disaccharides. Then you have performed the Seliwanoff's test, which is used to differentiate the keto and aldose sugars. Then you have done whether the given solution is polysaccharide or not. For that, you have used the iodine test. Now you have performed the Bial test to find out whether the given solution is a pentose or not. Okay. So, if it is pentose, usually it is a ribose. Where ribose exists usually? It is present in RNA. Okay, ribonucleic acid. Right. So, on a hydrolysis, RNA gives the ribose. Then, so now it is clear, but you know that you have disaccharides. So, disaccharides are, you have 3 different disaccharides here. You have maltose, you have lactose and you have sucrose. Okay, 3 different disaccharides are there: maltose, lactose and sucrose. This is lactose and this is sucrose. Okay, now how can you distinguish these 3 disaccharides? Okay, so for that we have to perform the Osazone test. Osazone test. So in Osazone test, what you have to do, you are using the reagent called phenylhydrazine. Phenylhydrazine and sodium acetate, glacial acetic acid. These 3 are the components of this reagent. So what you have to do, you have to take the test solution, to that 10 drops of glacial acetic acid is added and it has to be mixed properly. Then you have to add 1 spatula of phenylhydrazine and for that again you have to add 2 spatula of sodium acetate. Okay. So 1 spatula of phenylhydrazine and 2 spatula of sodium acetate. Then you have to mix properly. If any impurities are there, you can filtrate it and then you have to keep it in boiling water bath. So for glucose, you have to boil for 5 to 10 minutes and for other sugars, that is disaccharides, you have to boil for 15 to 20 minutes. Okay. After boiling, you have to cool it slowly, not immediately in the running water, you have to cool slowly. Thereby you can observe the formation of crystals. So these crystals can be observed under the light microscope in 40x, that is 400 magnification. There you can observe the clear crystal shapes. Okay. So here we have the picture for that, you please go through. Right. So this is the first one, which shows the bundle of grass crystals for glucose. Glucosazone, if glucose reacts with phenylhydrazine in presence of glacial acetic acid and sodium acetate, it gives the bundle of grass crystals which are due to glucosazone. Next one is galactosazone. Galactosazone crystals are needle shaped. These are needle shaped and for maltose you can observe sunflower shaped crystals. And for lactose you can observe powder puff or hedgehog puff. Okay, you know powder puff, how it exists. So or hedgehog shaped type of crystals. Okay. So why this reaction is, what is the principle behind this? Okay. So where this phenylhydrazine will react. So you can see this equation. So this is phenylhydrazine, it reacts with sugar, that is nothing but here you have the glucose. When it reacts with glucose, first 2 carbons are involved in the reaction, first 2 carbons. Thereby you can form the glucosazone. Here you can see the glucosazone. So in the same way, as you know that glucose, fructose, mannose. The 3 carbohydrates are differentiated only in the first 2 carbons. But here as the first 2 carbons are involved in the reaction, so the all glucosazone, otherwise fructosazone or mannosazone, 3 are of similar type. And they will be exist like this. Okay, bundle of grass crystals. So we have already done that one. So here you can see this is the after boiling and cooling you can observe the crystals. Okay. So these crystals has to be poured in watch glass and then they have to be transferred onto the slide, and this slide can be observed under the 400 magnification. Okay, that is 40x magnification. That is. So this is the galactosazone crystals, glucosazone crystals. Okay, what is the shape of this? This is the bundle of grass. So you can observe under the microscope. One by one, go and observe under the microscope. Students, you have observed the Osazone test. Now, so finally we are concluding. So what we have performed till now, first of all, to the given solution we have done the Molisch test. So, if the in Molisch test if there is no reaction it indicates non-carbohydrate. If you observe the brown color or purple color ring at the junction of two liquids it indicates it is a carbohydrate. Then what you have done, you have performed with iodine. Okay. So, when you done with iodine, if it is there is no color, then it indicates it is a simpler carbohydrate, mono or disaccharide, and if it gives blue or black color complex, it indicates it is a starch. Then you went for this Barfoed's test. So, in Barfoed's test, so if there is red precipitate within 1 minute or 2 to 3 minutes, it is a reducing monosaccharide. If on prolonged heating, if the precipitate is observed, then it is a disaccharide. Then, so afterwards you have done the Seliwanoff's test. In the Seliwanoff's test, if you observe the blue that is cherry red color product, then it indicates it is a keto sugar. If there is no reaction with Seliwanoff's, then we can go for we can conclude that it is a aldose sugar. Then we have performed the Bial's test. So, in Bial's test if you can observe the formation of green color product, it indicates it is a pentose, and if there is no reaction, then it indicates it is a hexose, right. So, like that you have to perform all these tests, okay, and you have to conclude what is the given carbohydrate. Okay. Thank you.